Preparing a gram stain. For this experiment you will need a slide dryer, an object slide, 2 inoculation loops, a Bunsen burner, a bacterial culture, tweezers, a squeezy bottle with saline, and a lighter. Make sure you have filled a colour series already and check that the demi-water is clean. If necessary, you will have to refresh the demi-water before you start the staining. If necessary, remove any grease from your slide with alcohol. Leave the slide to dry thoroughly before beginning the gram stain. Label the slide by writing on the matte surface. Use a pencil as it won't dissolve in the alcohol. Think about writing your initials, class, date, and the type of bacteria used. Put a small droplet of saline on the slide using a squeezy bottle. Flame sterilize an inoculation loop and take a sample of bacterial culture, preferably a single colony. Be sure not to take too much material, just touching a colony gives enough cells for a stain. Place the inoculation loop on the slide next to the droplet of saline and spread the bacteria on the slide. Take a little saline from the droplet and mix it into the bacteria. To properly suspend the cells, you need to repeat this step about 3 times. When you have an even suspension, mix the rest of the saline so that there is both a concentrated and less concentrated part on the slide. To mix, bring the loop from the cells that have been spread on the slide into the saline and mix the suspension in the droplet. Flame sterilize the inoculation loop after use. It is also possible to make a gram stain from liquid culture instead of from a colony. In this case, you don't have to resuspend the bacteria, but can start from the following step. Now let this slide air dry at room temperature or on a slide dryer at about 45 degrees Celsius. When the slide is dry, the bacteria must be fixed to the slide. This is done by gently passing the slide through a blue flame 3 times. This kills the bacteria and fixes them to the slide. The heat also denatures the proteins, which helps the cells to stain better. Before beginning the staining, allow the slide to cool down. Use a tweezer to place the slides in the colour series. Place the slide in the container with crystal violet for 1 minute. After this, wash the slide well with demi-water and let the water run off the slide. When the container with demi-water becomes very purple, it can be replaced with fresh demi-water at any time. Now place the slide into lugnol solution for 1 minute. Remove the slide and let the excess lugnol solution drip off and give the slide a short wash in 96% alcohol. Now place the slide in the second container with 96% alcohol for 1 minute. Wash the slide well in demi-water and let the excess water drip off. Again, you can refresh the demi-water if necessary. Now place the slide in the safranin for 1 minute. Wash the slide well in the demi-water and let the excess stain wash off the slide by holding the slide vertically and dry the back of the slide. Dry the slide at room temperature or on a slide dryer.